Our institute is working on marine microbiology, and we're specifically interested in the diversity of microorganisms that is out there in the ocean and the processes that they regulate in the ocean. So microorganisms play a crucial role in regulating, amongst others, our climate, like the uptake of carbon dioxide from the atmosphere is regulated by algae mainly, and the breakdown of this algal biomass is on the other hand. So the release of CO2 from this algal biomass is again regulated by the activity of microorganisms. So very important for the research that we do is mass spectrometry, and we have various mass spectrometers in our institute to measure actually the uptake of labeled substrates by microorganisms. And where I can show you some of these instruments. Here on this one in particular, we combust actually organic matter, and we determine the isotopic signatures of this organic matter, and we look for carbon and nitrogen isotopes. So this is for analyzing gases, and the mass spec is actually standing over here. And then on that side we have an elemental analyzer that actually does nothing else than combusting organic matter, turning it into CO2 and nitrogen gas, and then we measure with this instrument the isotopes of that nitrogen gas of the carbon. My interests were always in nature. I mean, as a young kid, I liked going out in nature, watching birds, for instance. But another thing that was a big hobby of mine is paleontology. I liked to collect fossils. So I went collecting fossils already as a small kid. Actually, at that time, I was not thinking that much of becoming a scientist. I was curious, but I was not yet it was not that clear yet for me what I wanted to do. And then I went over a HAVO, ended up on a laboratory school, and at that time I was convinced that I wanted to become a chemical laboratory assistant. I have a strong interest in chemistry, but what I was missing is actually going outside the laboratory. We have quite a few of these radioactive labs. I mean, like, we can go inside, but it's not dangerous amounts of radioactivity. Here we purified substrates that are radioactively labeled. Then we can analyze actually how much radioactivity is in there in another room in a so-called scintillation counter. And it gives very accurate measurements of uptake of substrates. I really wanted to go into the field to combine my passions, and that was like going into nature, going out into the field with my profession. And that is actually when I found a PhD position at the Netherlands Institute of Sea Research. That was there. They had a position at that time that was described as studying Cretaceous, so-called Cretaceous oceanic anoxic events, looking at which conditions led to the deposition of oil shales. So I recognized myself in that, and actually I never believed that something like that would be possible. So I did my PhD there and finally did indeed make it out into the field collecting samples. The latest instrument that we got in our institute that allows us actually to determine the isotopic composition of a single cell, and that's something that only this instrument can do. And with that we can get information about the activity of organisms. Of the earlier runs, I'm curious what came out. One of the major discoveries was that there was, at the time that I started working, a relatively clear picture of how the nitrogen cycle works in the ocean. And nitrogen plays a crucial role actually in algal growth, because algae are limited by the availability of nutrients, and the biggest part of the ocean, at least 70% of the ocean, it is actually the availability of nitrogen that limits their growth. The processes that also lead to the removal of nitrogen, and that's what actually have been mainly working on, that lead to the loss of this nitrogen, were believed to be actually only one process. That's a so-called heterotrophic denitrification. That is where organisms break down organic matter by respiring nitrate. And it was believed that this process was the sink for fixed nitrogen in the ocean. And that's where I started. But by that time that I started here, there had just been a new process discovered, and actually in the Netherlands, in a wastewater treatment plant, and that's the anaerobic ammonium oxidation. And that process is not done by organisms that respire nitrate to break down organic matter, but these organisms actually respire nitrite to oxidize ammonium. It was generally believed that these organisms grow too slow because they divide once every 14 days, that they could not be important in the ocean. We could show actually that these organisms might be actually the main sink for fixed nitrogen in the ocean. So this completely changed our view of how the nutrient availability in the ocean is regulated, how the key nutrient, nitrogen, is actually being regulated by microorganisms. Although I did my PhD at the Netherlands Institute for Sea Research, I actually didn't go to sea there, except for taking the ferry from Den Helder to Texel on a regular basis. But I didn't go for cruises. One of the most memorable moments was actually when I came to the MPI in 2001. At the end of that year, over Christmas, there was a cruise going to the Black Sea. And I was fortunate enough that actually this cruise was going over Christmas, because they had already planned in the whole crew for this cruise already a long time before, but when it turned out that it was a Christmas cruise, there were not so many people anymore that were enthusiastic about it. So some places became available, and actually I joined this cruise, and this is actually the cruise that also led to the discovery of the first anaerobic ammonium oxidizers in the environment, where we could show for the first time that these organisms in the world's largest anoxic basin, the Black Sea, that these organisms were responsible for anaerobic ammonium oxidation. And it started out with a big storm, so all of these, it was a real ocean-going mission, and it was a very special experience.